Hi, welcome in everyone to this PHA Live webinar. We will begin shortly. Thank you for joining us this afternoon. We appreciate everyone taking their time out to spend some time with PHA and share this important information on PHA research, the spotlight on innovative projects in PH. Before we begin, actually, I would like to take a moment to thank our PHA Connects program sponsors. Sponsors do not participate in the development of and do not endorse the content of programming. We appreciate their support. Sorry about that. We also like to take a moment to invite you to become a member of PHA, joining the Pulmonary Hypertension Association's, the world's largest organization focused on raising PH awareness, empowering and supporting patients and their families, bringing meaningful connections, and more importantly, for one day finding a cure. So we welcome you to reach out to us at memberships@phassociation.org if you would like more information on how to be a member. If you are a member, thank you so much for your support. A few programming notes before we get started. We will have Q&A not only following our presentation, but our presenters will be able to answer questions specifically about their portions of the call. If you do have a question, please use the toolbar, the Q&A toolbar, to submit those questions to our panelists. And then immediately following the webinar, so once I close it out for the afternoon, there will be a survey. If you would take a minute or two to complete that survey and let us know what you would like to see in the future, this will help our programming and making sure that we bring you webinars that you are interested in and that are sharing the latest information in PH. And now I would love to welcome Dr. Matthew Lammy, who will serve as our moderator for today's call. He is an associate professor of medicine at Johns Hopkins University, as well as the current chair of the research committee here at PHA. So we welcome him and thank him for moderating this call with our lovely panel of speakers who will present their research and the awards they received from PHA. So Dr. Lammy, thank you, and I will allow you to take it away.
All right, thank you so much. We're all very excited to be here. We have 4 people on our panel today, all of whom had received PHA awards that have funded their research. You'll see that they're at various stages of their career, but are all doing very exciting and innovative work. As Angela mentioned, we'll have a Q&A after each presentation, as well as a wrap-up at the end with more questions, and we'll be monitoring the Q&A to make sure that questions get addressed. And just a reminder that we're really going to focus on 2 types of questions today. Number 1 is related to the research of each individual investigator, and also how PHA has helped support them through their mission. Unfortunately, we don't have the time, or this isn't the proper forum to answer questions about individual cases or medications. So we're trying to keep things focused on the science, and but we welcome all questions related to that. So without anything further, I'd like to introduce Dr. Adam Andruska, who's an instructor at Stanford School of Medicine, and he'll be talking about T-cell vascular signaling as a therapeutic target in PAH.
Can you hear me okay? Thank you, Dr. Lammy, for the kind introduction. I'm excited to take everyone through some of the studies we're doing looking at how the immune system contributes to pulmonary vascular disease. And so I just want to start, pulmonary hypertension is a disease of the small arteries of the lung. And to illustrate this, I just have a stain here of a normal human lung where you can see a pulmonary artery here on the top adjacent to an airway. And as you can see, this is a somewhat thin artery that's very compliant, has several layers to it, an endothelial layer, a muscle layer, smooth muscle, medial layer, and then an adventitial layer on the outside. And in pulmonary hypertension, what we see is the changes you see here, where kind of all the layers are thickened and hypertrophied, but particularly there's a development of an interesting cell type we've been studying called neointima that forms between the endothelial layer and the smooth muscle layer, and it occludes the vessel lumen. In addition, we see infiltration of immune cells, and all this contributes to resist the flow of blood through the lungs, and that results in form of right heart failure, as illustrated here from a patient who underwent heart-lung transplant at our center. And we've been also interested in how the immune cells, the immune response around the blood vessels might be contributing to this. It's long been recognized that there are substantial lymphoid or white blood cell aggregates made up of what are called T and B cells around the blood vessels. And we became interested in this because we've developed a mouse model where we give a mouse a chronic inhaled antigen daily for weeks. And what we see in the model is that perivascular inflammation develops around the blood vessel, similar to what we saw in the human disease. And then the media, the smooth muscle hypertrophies, and in around 4 weeks, we see development of neointima cells, which go on to occlude the vascular lumen and give rise to pulmonary hypertension. And we notice that the lymphocytes, the white blood cells here in green and pink, seem to be very prominent during this process. And further, the structure of the immune response seemed to match what we would see in human disease too, where here's our mouse on the left, and you can see these green smooth muscle cells occluding the blood vessel lumen with the white blood cells in red and pink here. And it looked somewhat similar. Zooming out, we would also find structures of immune cells denoted by these arrows here, known as follicles. So collections of T and B cells that were forming where the blood vessel was remodeling. And that reminded us of certain forms of severe, this is drug and toxin-induced pulmonary hypertension, where we see these aggregates of white blood cells right adjacent to the blood vessels. And a very thick section of mouse lung stained for smooth muscle cells in green, as well as inflammatory cells in red and other vascular markers. And if we zoom in on a small area of this and kind of look, you can see an airway here that are green smooth muscle cells, and then a pulmonary artery. And this particular pulmonary artery is remodeled. And you can kind of see as we rotate it around, it's occluded by smooth muscle cells, just like we saw before. But what was interesting to us is that the immune cells, when you add those in white, completely envelop this area where the vessel is remodeling. And so we're really interested to see how these two kind of environments are communicating to one another and how that might contribute to the development of pulmonary hypertension. Now, we needed a place to start. We needed a cell to kind of focus in on. And so what we did is a series of experiments where we stained the immune cells in the lung with a panel of 21 different antibodies that mark different immune cells. And so this is just a schematic of the lung tissue that we stain. And the power in doing this is that we get a lot of information about the different immune cell types in the lung because of the 21 different markers. But then we also understand where each cell is coming from because it's a spatial technology. And so we can narrow in on just the area around the blood vessel, and that's kind of illustrated here for the staining patterns that we showed for some select markers, and essentially just count up the cells that are around the different blood vessels. And what we found is that early in the disease, T cells seem to be very prominent here in the cyan. And that really drew our attention because that was what we were also seeing in the histology previously. So we went on to validate this. And since we're using a mouse model, we can use different technologies to kind of probe our hypotheses. And so we used a mouse model that has all immune cells except for T cells in it. And what we found is under these conditions, the mouse would not form neointima, pulmonary hypertension, or this organized immune response outside the blood vessels. But then what we did is we transplanted in normal T cells back into the T cell deficient mouse, and it completely recovered the phenotype, meaning that we got the immune structures back, the neointima reformed, and we also found the development of pulmonary hypertension. And so to us, that really focused us in on T cells as a critical cell type in the immune response, coordinating, a transition from this normal kind of thin compliant pulmonary artery to this remodeled pulmonary artery with this complex immune response around it. This was also interesting to us because T cells are implicated, T cells are not a homogeneous group. It's a family of different, complex family members, and there's good actors and bad actors. And there's been studies to show that there are regulatory T cells, which are very helpful in preventing pulmonary hypertension, whereas there are certain bad actors that have been implicated in specifically schistosomiasis and hypoxia-induced PH. So that led us to the hypothesis that there exist pathologic T cell subsets, so sub-family members that may be driving the development of neointim development. And if we can identify and target these cells, it would be relevant for treating human disease. And so what we want to ask, the questions we're asking with the help of the PHA is, can we precisely identify these T cell subsets, and what are the signals that these cells are signaling out, and can we interfere with them? And then we're also looking at how they coordinate the immune response, but that's a little bit bigger talk than what I'll cover today. So I just wanted to illustrate a couple of our studies that the PHA is helping us with. And one of the big ones is we've been doing single cell RNA sequencing of our mouse model under different experimental conditions. And so in this case, we dissociate out the mouse lung into individual cells, and we send each cell off for sequencing. We also do some antibody studies and things like that at the same time. But essentially, we get a readout of all the genes for each cell in our model. And here, this is a graphic illustration of this, of about 25,000 cells from different conditions. And we use the gene expression to cluster or group the cells together. And that's shown in terms of the groupings and the colors here. And then we give them annotation. So we have our T cells and B cells and others, and as well as vascular cells. And then we look across our experimental conditions and we see how do these populations change in different conditions. And so we are looking at inflammatory conditions that don't form neointima or pH versus those that do. And what's exciting to us is we've been able to identify populations of T cells within our grouping here that are specifically upregulated while neointima forms. And they've also been implicated in other autoimmune and inflammatory diseases such as multiple sclerosis, rheumatoid arthritis, certain hypersensitivity reactions. And so we're really interested in using the gene profile of these cells to really finely characterize them and see what agents are available that we could use to specifically target these cells and potentially treat the immune response specific to neointima. The other thing that we're doing is building signaling maps, cell-cell signaling maps using our RNA sequencing. And here we use the RNA-seq data, and essentially we're trying to build an atlas that says how one cell signals to another based off of the expression of the signaling ligand and then the receptor of the receiving cells. And so we take our RNA-seq data and we've made these, they look like kind of complicated wire diagrams of cell types at the top signaling down to cell types at the bottom, and we can assign signaling strength. But probably this is an easier diagram to look at where just specifically for T cells, we've been able to kind of map out in these pie charts different groups of signaling axes that we think are going from T cells to different components in the lung vasculature. And then we try to prioritize those based off of strength, and then go on to see how they affect the formation of neointima in our model. To do this, though, we don't just rely on the single cell data. We also go back to the human tissue. And so once we identify different signaling and receptor candidates, we first go into our mouse model and we see how are those genes actually being expressed around the blood vessel. And so this is a particular signaling molecule that we're very interested in, that's shown here in pink, that seems to be really highly expressed in the T cells around the vessel, as we kind of predicted, whereas the receptor is kind of all throughout the vessel and other cell types. And what we were excited to see is when we went to human pulmonary hypertension, we could also find this signal being expressed in cells kind of around this plexiform lesion here and other lesions shown here on the right, I mean. And we've found that these cells are indeed T cells through other studies, and we've validated the receptors. And so for this signaling axis, we've gone on to go, we predicted in our mouse model, we validated in human tissue, And then we've been able to develop and find a monoclonal antibody that specifically targets this signaling axis. And we begin to test this in our mouse model. And we found that it does a reasonably good job of preventing the formation of neointima in our model, as well as dampening pulmonary hypertension. And we have more studies undergoing to kind of build this up. We're also looking at the regression of pulmonary hypertension using this monoclonal antibody. But we think it's interesting because we're not really ablating immune cells to achieve this. We're not, using rituximab or Cytoxan to delete whole immune populations. We're mainly just disrupting a single signaling axis in the immune response and showing that that's sufficient to disrupt the formation of neointima and prevent pulmonary hypertension from forming. And so we're really excited about this particular target and moving it forward. So that is essentially what we're working on in a nutshell. And so hopefully I've maybe convinced you that, neointima we think is not only an important feature of group 1 pulmonary hypertension. These are all specimens from our advanced lung disease transplant program that we've banked, but you also see neointima in other forms of pulmonary hypertension, 2, 3, and others. And so if we can target the formation of these cells, we think it would be, hopefully broadly applicable to a wider group of patients. We also have a line of research to say that we believe that T cells are important for the formation of neointima in this model, and that maybe disrupting these pathologic T cells are a way forward for treating PH. And we're also specifically interested in looking at other forms of inflammatory lung diseases, particularly where there's crossover between, say, interstitial lung disease and pulmonary hypertension, like in scleroderma, to see if some of the signaling targets that we are identifying are applicable there. And so thank you for your attention, and I really thank you for the forum to present all this, and happy to take any questions. And I have to acknowledge all my co-lab mentors and funding sources here.
Beautiful. That was a wonderful presentation, and I think a great example of using information from humans as well as doing lab studies and trying to put them together. So we invite anyone that has questions to put them within the Q&A. I also would like to invite the other panelists to ask questions to each other if they have them. But I'll get started with a question to get things rolling. In addition to having subsets of T cells to understand neointima formation in pulmonary arterial hypertension, do you think it's possible that these T cell subsets could be used as biomarkers for determining disease severity? So using them in blood samples, for example, to determine how severe the pulmonary hypertension may be?
Yeah, thank you. I think it's a great question. We're always really interested in kind of contrasting what's going on in the lung to what's going on in the peripheral blood. And, Andy Swed and Grzesia Kłapeczewski in Austria, they've all been very much interested in, cytokine profiles and things like that. I think that what we'd ideally like to do is kind of move to where we can take lung samples, which we are doing, and dissociate those and do the single cell on the T cells themselves, and then compare it to what we're getting in peripheral blood. And there is a mechanism for doing that here at Stanford. So yeah, that is something we're very excited in. And I think, that correlation would be very important, because I would imagine things get maybe noisier as we move out to the periphery. And if we can, see how things over a broad panel kind of correlate lung versus periphery, I think that would be very helpful for, identifying who should be targeted for maybe immune therapies versus other therapies, and perhaps even prognostication.
That's great. One other question I had while we're waiting for others to come through, and again, we'll have a separate session at the end if there's residual questions. Just briefly, it looks like, as you showed, T cells may be more important in the earlier phases of disease development, and then B cells seem more important later. I know that it was probably beyond the scope of your talk, but do you have interest in looking at B cells in sort of established disease? Because if we're looking for treatments for people that are living with pulmonary hypertension, figuring out what immune modifiers you can use at that point seems to be most relevant.
Yeah, no, that's a great question and super relevant for all of our connective tissue disease patients as well. Yeah, I think we took a very broad approach of just new intima formation to start off with, and we are trying to move out and more finely look at, okay, we get disease established, and if we take away the immune driver and things resolve, what are the signals that are going on there? There's a lot of literature to say that T cells are required later in disease to kind of promote survival signals to, mature B cells into the plasma cells that are making, antibodies and so yes, it may move from T cell to B cell, and we are open to that kind of like line of experimentation. It's a big area. But I think there might also be some further things to learn from the T cells just on the survival signals that they send out to the B cells to potentially target those which may be relevant for late stage disease. But yeah, great question. I think the B cells are a huge part of this.
Okay, great. Just to make sure everybody has equal time, we'll thank you, and we'll move on to the next presentation, which will be by Dr. Sadaya, who is an associate professor of pediatrics. She's the 2023 PHA Early Career Mentored Scientist Award winner, and she'll be talking about non-invasive predictive markers of pulmonary hypertension associated with bronchopulmonary dysplasia in preterm infants.
Okay, thank you everyone for joining us today, and thank you for this great opportunity to present our work. These are the funding sources that has supported my research, and I would like to thank PHA again for the funding available. I'll be switching the gears a little bit here and talk about pulmonary hypertension in developmental lung disease context, specifically in those who are born prematurely. With improved obstetrical and neonatal care, there has been improved survival in extremely premature infants. And this results in babies who are born with lungs that are still in developmental stages or the saccular stages, who are then exposed to noxious stimuli, resulting in pulmonary complications. And most commonly, this is called as bronchopulmonary dysplasia or chronic lung disease of prematurity. In addition to abnormal growth of alveolar sac or air sacs, there is also abnormal development of the blood vessels in the lungs or the pulmonary vasculature, which results in reduced number of blood vessels, reduced density of the blood vessels in the lungs, altered vasoreactivity, high vascular tone, and smooth muscle proliferation. All of this resulting in the more severe complication of prematurity, which is pulmonary hypertension, and is associated with high mortality and morbidity. I will be referencing to this as BPD-PH through the rest of my presentation. Clinically, diagnosing pulmonary hypertension or BPD-PH is a challenge, and this is mostly due to the overlapping clinical signs and symptoms with that of BPD or just a chronic lung disease. The gold standard for diagnosing BPD-PH is through cardiac catheterization, which you can imagine in these infants who often weigh less than 4 pounds is highly invasive and carries a lot of morbidity. So the current clinical practice is to use echocardiogram for diagnosis and management by studying the stress on the right side of the heart, and is often difficult in these infants who have significant hyperinflation of their lungs due to BPD and are often agitated and don't have a very long window of getting these echocardiogram images. Pathophysiology of BPD is varied and complex. It comprises of a combination of antenatal and perinatal insults along with genetic and epigenetic factors, which leads to abnormal development of the lung circulation and eventually pulmonary hypertension. Given the complexity of this pathophysiology, identifying a single biomarker for this pathology is a challenge. We approached this challenge with an unbiased approach, doing a multi-omic research to identify a molecular signature or an endotype in the tracheal aspirate, which is the lung lining fluid, in preterm infants with BPD-PH. We further identified the underlying target gene expression and associated pathways through an in silico model. We identified a panel of mRNA gene transcripts, proteins, and microRNAs, and the in silico predictive modeling identified VEGF, TGF-beta as some of the key signaling pathways, which also serves as key transcription factors for angiogenesis in neonatal lungs. And of particular interest are the microRNAs, which are small non-coding RNA and play an important role in lung organogenesis by regulating various gene expressions. Scientists Victor Ambrose and Gary Rufkin received Nobel Prize this year for the discovery of microRNAs and its role in post-transcriptional gene regulation in the 1990s. So of the 12 microRNAs that were differentially expressed in the infants with BPD-PH compared to those with BPD alone, of particular interest was microRNA 29A, which has been studied in pulmonary hypertension and sexual dimorphism in various human and animal models. And this was the microRNA that had the highest average expression and also highest differential expression in the samples. Early identification of these markers could predict development of BPD-PH in preterm infants and hence help improve outcomes. For this, we conducted a prospective study where we recruited babies who were admitted to the neonatal care unit who were born extremely premature with less than 28 weeks of gestation, who also needed mechanical ventilation at 7 days of age when we collected the tracheal aspirate samples. And we followed these infants prospectively. At 36 weeks, they all receive a screening echocardiogram to diagnose BPD-PH. And of these 33 infants, 15 had that diagnosis of BPD at 36 weeks and 18 with BPD-PH. We further conducted logistic regression analysis to calculate the predictive value of the 12 microRNAs. Our analysis showed a receiver operating characteristic area under curve of 0.89 with sensitivity and specificity of 77% and 81% respectively, which is a great predictive value. However, we had numerous challenges to advance our study further. While tracheal aspirates is a great way to obtain a non-invasive biofluid, it can only be obtained from those infants who are mechanically ventilated and are intubated, which also skews a sampling population to the more sick infants in the neonatal ICU. And also, once the infants are have clinically improved, they get extubated, which limits our collection of future longitudinal tracheal aspirate samples, and in general, limits the number of babies we can include in our study. Hence, we explored saliva as an alternative non-invasive biofluid. We did conduct a preliminary study comparing tracheal aspirate and saliva samples and examined the microRNA profile in both the biofluids. Of the 161 microRNAs had similar expression in both tracheal aspirate and saliva, as seen in the scatter plot here. With this information, we conducted a similar prospective longitudinal study recruiting 71 premature babies in the neonatal ICU. We collected saliva samples at 7 days of age and followed them prospectively until 36 weeks post-menstrual age, when they typically get their echocardiogram screening for BPD-PH. We had almost twice the number of babies, since it is easier to collect the saliva samples, and 36 of them had BPD, while 26 of these infants developed BPD-PH. And all of our infants tolerated collecting these saliva samples very well, which, as you can see in this image, uses a collecting kit with a small swab at the tip, which is introduced gently in their mouth and left there for 30 seconds. There were no adverse effects. We then calculated the logistic regression analysis for predictive value of the 12 microRNAs. And this time the ROC area under the curve value was 0.94 with a sensitivity of 93% and specificity of 78%, which was comparable or even better than that for the tracheal aspirate. We further conducted another preliminary study to explore the mechanistic pathway of the microRNA 29A in angiogenesis pathways in pulmonary artery smooth muscle cells, which are hypertrophied in BPD-PH. We transfected pulmonary artery smooth muscle cells with microRNA 29A and further studied various gene expressions through that we had identified in the in silico model. And our study showed that the PDGFA was significantly downregulated in those cells that were transfected with microRNA 29A compared to those which were control samples. And PDGFA is one of the key drivers of angiogenesis in neonatal lung and could be the key signaling pathway in BPD pathogenesis, which warrants further investigation. In summary, we have identified a molecular signature of BPD-PH in non-invasive biofluids, such as including tracheal aspirate and saliva. This panel of microRNAs could potentially identify high-risk preterm infants for developing BPD-PH, and we can identify them as early as 7 days of age. Saliva is a promising and conveniently accessible biofluid for conducting non-invasive biomarker studies, especially in high-risk preterm infants. With this, I would like to thank all the members in the BPD research team, my mentors, and the Penn State Children's Hospital Pediatric Department, and the core lab for its support. Thank you.
All right, that was wonderful. Thank you so much. And as you can see, PHA is interested not only in adult PH, but also pediatric pulmonary hypertension, which I think is kind of an underfunded area generally in this country. So lots of thanks for focusing on these very sensitive patients. While we wait for any questions to come through in the Q&A, I'll just start out with one. It's exciting to see the studies using saliva from preterm infants to detect the miRNA biomarker panels for BPD-PH. How large do you think would follow-up studies need to be so that we can start to move this towards clinical applicability, FDA approval for diagnostic purposes, and like what would be the pathway to take your very exciting, compelling data and moving that to actual clinical use?
Thank you. That's a great question, since while BPD-PH is a very important pathology to be looked into, it is not, in terms of N, we don't have as many patients as, say, for example, COPD or asthma, which limits the number of patients that we can enroll for a clinical study. Having said that, our next goal would be to conduct a multi-center study so we can validate our results in other population in other institutions before we can move on to a clinical trial.
Yeah, exciting that that you're pushing this forward, and I think the PHA has been involved in the process as well. One question from the Q&A, which is, what cutoffs are you using to diagnose BPD-PH using echo in these infants? And can you talk about any sort of limitations to that as the gold standard for diagnosis?
Yeah, so in our study, we have used flattening of the interventricular septum, TAPC, right ventricular dilatation, and hypertrophy as the markers for pH. In terms of the limitation, I can go on for the rest of the hour. Studies have shown that the markers for on the echocardiogram are pretty sensitive in detecting pH in general. However, the challenge is in this patient population where often the babies are, if they are not comfortable, they will be agitated, there'll be a lot of movement. And also, as we can imagine, the babies are small and they have a very narrow window to visualize and have optimal views on the echocardiogram, which again limits, and we need a very experienced technician to capture the windows that we would ideally need for getting all the parameters. So hence often we have incomplete windows, especially for the TRV, that again limits calculation of the right-sided pressure. So there are numerous challenges, and on top of it, having a pH expert cardiologist who is reporting on these echocardiograms, have the expertise in the pH, and is evaluating all the needed cardiac parameters is also key to optimize this as a tool.
Great. Well, thank you for tackling all those challenges to push your work forward. Again, if people have further questions, please put them in the Q&A and we'll try to address them at the end. Next, I'd like to introduce Dr. Yuan, who'll be talking about pericytes and their contributions to pulmonary vascular remodeling. She has been awarded 3 different PHA awards, it seems like every 4 years, starting in 2015. And I think is a nice example of how PHA funds sort of early career investigators, and now her most recent award is an innovation award, which is open to investigators of all levels. So we're excited to see her presentation. Dr. Yuan, it looks like your screen size is a bit off from what I'm seeing. It is larger, I think, than you want it to be.
I can see it. I'm sorry, this is Angela. I can see it. I'm not sure.
It must just be me. All right, if you can see it, then please.
It looks
please.
good to me too.
All right,
Okay.
it's just
Definitely got nervous when the slide doesn't work. Well, thank you so much, Dr. Lamy, for the kind introduction. I'm very excited to be here and share the ongoing projects on pericytes in pH that are supported by PHA. Pulmonary arterial hypertension is a rare disorder that characterized by progressive elevation in the pulmonary pressure that, if untreated, leads to right heart failure and death. A combination of endothelial dysfunction, proliferation of smooth muscle cells, and in-cell thrombosis leads to progressive narrowing of the blood vessels and eventually leads to right ventricular hypertrophy. In the US, the estimated incidence and prevalence of PH is nearly 12 cases per million adults. PH can develop in men or women at any age, but it is most common in women, as the female to male ratio is 4:1. Current therapies include vasodilators, but despite its overall effectiveness, they failed to stop progression of the disease, leaving the lung transplantation with its inherited complications and high mortality as the only option available for end-stage patients. Therefore, there are always unmet clinical needs for new therapies. Here, I'm showing you the contrast image of the lung vasculature in PH versus healthy. The distal portion of the vessels disappeared and leaving behind the only main branches. Let's take a closer look at what happened to the vasculature. The schematic image on the right shows the abnormalities throughout the pulmonary circulation. There are muscularization and loss of small precapillary arteries. The new intima formation occlude the median-sized vessels. And lastly, the plexiform lesion formed in the larger-sized vessels. The histological images are showing the lumen is minimum or occluded. This resulting in the high pressure. My lab has been dedicated to study the early stage of the remodeling process and how pericytes dysfunction leads to the local capillary remodeling and exacerbates subsequent upstream alveoli remodeling in PAH. In order to form functional vessels, one of the key components is pericytes. They are mesenchymal-derived mural cells that wrap around endothelial cells of capillaries throughout the body. They are embedded within basement membrane that direct contact with endothelial cells. Their presence is required for capillary stabilization, maturation, and remodeling. To study pericyte function in idiopathic PAH patient samples, we reanalyzed the single-cell RNA sequencing data from a published work. Among 30,000 cells, both endothelial and mural cell population are identified from 8 control and 3 IPH samples. We further extract mural cells and endothelial cells and recluster them. Based on the markers we previously published, for pericyte, including CSPG4, PTGI receptor beta, COX4I2, and HIGD1B, we identify pericyte cluster precisely. Intriguingly, when we applied pseudotime analysis, we found control pericytes a mixture of color, suggesting an active and multipotent stage, whereas IPH pericytes are purple, that indicating a inactive and fully differentiated stage. In the literature, pericytes has been described as multipotent stem cell-like, suggesting their lineage can become other cell types under different stimulations, including fibroblasts, myofibroblasts, adipocytes, osteoblasts, and the lineage of our interest is vascular smooth muscle cells. Using a 3G5 antibody, we can precisely label the pericytes in capillary nets in the healthy human lungs by those white arrowheads. When we stain them in IPH lungs, the occluded blood vessels co-express smooth muscle actin and 3G5, suggesting pericyte may become smooth muscle-like and a part of the vessel lesions. Pericyte for a long time has been controversial because they are shared markers with smooth muscle cells. Pericytes are positive for NG2 and PTGI receptor beta staining. On the very top left, that's a black 6 mouse retina. Smooth muscles are in blue by smooth muscle actin, endothelials in green by isolactin-4, and the pericytes are red by the NG2 staining. You could tell the typical oval cell body with long processes covering the capillary endothelium. By utilizing Krelok's P system, we can label pericytes by promoter-driven reporter mirror line. For example, the pericytes are in TD tomato color, shown in the brain vasculature and in the heart vasculature. And those pericytes are negative for smooth muscle actin. And showing you in the long slices, TD tomato labels their oval cell body and the lung processes. Using reconstructed confocal images of the mouse lung, you could clearly find these TD tomato cells label pericytes in the capillary nets. The most commonly used mouse model for study pulmonary hypertension is the chronic hypoxic model. The mice are housed in 10% oxygen chamber for 3 weeks. This model is useful because it's predictable and reproducible. And the black 6 strain will have increased pulmonary arterial pressure and right ventricular hypertrophy. As shown here, the traditional H&E staining, the distal arteries in the red circles associate with some modest degree of vascular remodeling after hypoxia, as some thickening in the smooth muscle layers. My lab developed a cool technology is to do tissue clearing and deep imaging. And you can see by about 3 days, the lung lobes are opaque and are now become transparent. So then we could image the entire vasculature in 3D, as shown here in videos. We also found using smooth muscle actin staining, we could find the expansion of smooth muscle cells at the distal lungs from 50 micron size down to the 20 micron size vessels, suggesting hypoxia indeed induced vascular remodeling. And our imaging tools are so cool, so you can visualize the whole organ in 3 dimensions without any regional bias. And you could also apply this technology to any organ of interest, including hearts and others. Well, going back to the lineage tracing, our next question is if pericyte contributing to hypoxia-induced vascular remodeling. So we utilize NG2-TDT animal for lineage tracing. In the control lungs, pericytes in TDT color are positive for NG2 PTJ receptor beta in capillaries, and they're negative for smooth muscle actin or myosin heavy chain, which is our mature smooth muscle markers. However, after 3 weeks hypoxia, something happened. These TDT labeled cells translocated from capillary to small positive arterioles, and they are also positive for smooth muscle myosin heavy chain. So here I'm showing you reconstructed 3D confocal vessels. You could clearly see these cells were in capillaries, and after 3-week hypoxia, they cover around the arteriole and express smooth muscle actin and other markers, suggesting they do contributing to hypoxia-induced vascular remodeling. So far, I've shown you that exposure to chronic hypoxia is mostly common in the mouse pulmonary hypertension model. However, other forms of pulmonary hypertension lack mouse models. For example, increased pulmonary blood flow leads to advanced vascular lesions. We're seen in patients with pulmonary arterial hypertension in congenital heart defects. Left lung pneumonectomy in mouse, which can induce increased blood flow and shear stress in the lung, is sufficient to induce intimal remodeling in the distal arteries. Utilizing this surgery tool and lineage tracing, we want to evaluate pericyte role in the induction of microvascular remodeling in response to increased flow after pneumonectomy. So NG2-TDT mice received the tamoxifen to induce pericyte label, then underwent pneumonectomy. We found these mice show modest pulmonary hypertension and a right ventricular hypertrophy after post-operation day 7, and sustained pulmonary hypertension up to 42 days. After 3 months, the hemodynamics were back to normal. When we harvest the lung, we traced the pericytes and applied computational tools to convert fluorescence color into 3D meshes. The results show an increased number of pericyte processes and surface area from post-operation day 7 to 42, suggesting their response to increased blood flow. To take one step further, we collaborate with Dr. Mark Puder, who is a surgeon in the Department of Surgery at Boston Children's Hospital. We generated a novel extended pneumonectomy model by removing both left and post-caval lobes. 95% of the mice survived, and after 14 days, these animals have higher pulmonary hypertension and a right ventricular hypertrophy with increased muscularization than left lung pneumonectomy only. how vascular cells driving this remodeling process and the molecular mechanism for different cell behavior are ongoing projects in my lab. So from today's presentation, I hope I convinced you pericytes are essential for pulmonary circulation. And single-cell RNA sequencing is a useful tool to identify cells and predict lineage trajectory. And my lab is also working on identify different subtypes of pericytes, and we're trying to review their function in the gene expression levels. Every single time when I look at those cells in the lung, they have their unique structure. They have their old cell body and the long processes, just like an octopus have tentacles covering the whole vasculature. We want to study the underlying mechanism for pericytes in pulmonary diseases. Our ultimate goal is restore pericyte normal function and design new therapeutics to treat pulmonary arterial hypertension. So with that, I want to thank my exceptional fellows, mentees, and students who are the driving force and make everyday work fun and enjoyable. I want to thank my collaborators for supporting me, PHBI for providing tissue and all the funding resources. I'm very grateful for Pulmonary Hypertension Association, their generosity for support my research programs throughout my career, particularly when I start my first faculty appointment 3 months before the pandemic shut down. Lastly, I want to thank the patient and families who remain strong and fight against pulmonary hypertension. You are the reasons we are here. Thank you so much for your attention.
All right, thank you for your work and that beautiful presentation. We do have one question that has come through. This one's about the imaging techniques, asking, could these imaging techniques that you demonstrated for your preclinical model be used for studying parasites and markers in humans as well? Is there a way to translate the imaging techniques to a larger scale to humans?
Yeah, absolutely. We are trying to optimize this protocol and apply to human lungs. And we have some preliminary data, but we also found some technical challenge because once the lung were taken out of the body, it's just we need to make a way to expand the structure. Otherwise, it just clumps together. So we're working on that, and we wish our technology can be applied to study human lung vasculature. And this clearing methods also being used in other studies. People use it to study lymphatic diseases. So it is going to be the trend in the future.
Oh, great. Well, that's wonderful. Thank you so much. I think in the interest of time, we will move on. But as you can see from Dr. Yuan's work, pulmonary hypertension is not just a disease of the arteries, and we're going to see that again with our next presentation, the role of bronchial circulation in PH by the 2022 PHA Innovation Research Awardee, Dr. Galambos.
Oh, we cannot hear you.
Yeah, okay. Okay, great. Can you hear me now?
Yes,
Hello?
yes, we can.
Awesome, thank you. Thank you, Dr. Lamy, for this nice introduction. And also thank you for the organizers and the sponsors to have this webinar. It's just fantastic talks already. And for sponsoring my research on the bronchial circulation. And one of the hindsight is going last is that very hard to top of these beautiful presentations, but one of the perks perhaps is that I don't have to introduce what pulmonary hypertension is and what the clinical classification of pulmonary hypertension, because this was done beautifully by the speakers spoke beforehand. And but what I thought, because I'm a board certified pathologist and trained, I would say I would like to just show how pulmonary hypertension looks like under the microscope. And we usually look at two type of vessels to identify pulmonary hypertension. That's one of the pulmonary arterial side. This is a normal pulmonary artery, but in pulmonary hypertension, they got muscularized and then scarred. You can see the scar tissue or intimal proliferation with an attenuated lumen. And sometimes it is so obliterated with this proliferated vascular structures, a so-called plexiform lesion that is a hallmark of a severe pulmonary hypertension. When you look at the pulmonary venous side, you see that normally the veins are very nice and thin, but in pulmonary hypertension, they become very prominent and thick, and using some special stains like here with trichrome and elastin combination, you can see that some of these are obliterated, which leads to the diagnosis or pulmonary veno-occlusive disease. In the past 20 or 30 years, tremendous research effort focused on solving the problem that originated either from pulmonary arterial vessels or pulmonary venous vessels to solve the problem of pulmonary hypertension. But as been presented earlier, the cure for pulmonary hypertension is still lacking. So we decided that we're going to focus on another circulation and to see whether this circulation has anything to do with pulmonary hypertension. And this is the bronchial circulation, which is a systemic circulation in the lung that has a really important function. It brings oxygen and nutrition to the airways as well as the pulmonary arteries. As you can see here, they send some nutritional vasculature to the pulmonary artery. Although in other systemic diseases like arteriosclerosis and other problems, these vessels are heavily studied. In pulmonary hypertension, it has been really understudied. At the histologic level, we don't really see the the bronchial vasculature unless they are pathologic, like here. They're dilated and filled with blood, and sometimes we can see the around the arteries as well. So you can imagine that the problem with this circulation and can translate into pulmonary arterial wall hypoxia, changed metabolism, and the developmental of hypermuscularization and neointima. So how do we study bronchial circulation? So this is an example of a technology with differential ink injection of the pulmonary arterial side and the bronchial artery side. This is from an infant who died of developmental lung disease from a intractable pulmonary hypertension that caused by this diffuse small arterial obstruction. So I'd like to just focus on the upper part. This is how basically, just let's look at how normally the blood flows in the lung. So the pulmonary arterial blood comes into the alveolar capillaries, the gas exchanges and go to the pulmonary veins and go to the left heart, and then the oxygenated blood is going to go to every cell of our body. But we hypothesize that in this case, there was connections between the pulmonary circulation and the bronchial circulation. So we further hypothesize that therefore the injected inks, like that's the green from pulmonary artery and then the blue from bronchial artery, will mix up and they will present in the wrong area as well as mostly in the microvessels of vasovasorum and peribronchial areas. So normally there is no connections between the pulmonary and bronchial circulation. So our hypothesis based on that we can identify novel interactions or shunt vessels between the pulmonary circulation and bronchial circulation. So when we looked at the histological pictures of this lung, then what we saw was that exactly what we hypothesized. The green ink that was injected in the pulmonary arteries showed up in the bronchial artery, and the blue ink that was injected in the bronchial artery showed up in the pulmonary artery. And then when we looked at the microvasculature on the bronchial circulation, that are the bronchial vessels, we could see both mixed ink, blue and green ink, around large airways as well as small airways. So these findings we confirmed by a state-of-the-art imaging technology in collaboration with Dr. Lundmark and Weinovich in Switzerland and Sweden, the so-called imaging technique using synchrotron imaging that allows to reconstruct pulmonary vasculature precisely in 3 dimensions. Then you can see here that we were able to reconstruct the pulmonary arteries here and also the bronchial circulation, and we can easily identify open anastomotic connections between the pulmonary and bronchial circulation. In addition, we performed studies with the help of precise 3-dimensional computerized reconstruction of pulmonary vasculature using human samples from patients with idiopathic pulmonary hypertension. These are adult patients. And then we found that not only there were connections between the pulmonary arteries and bronchial arteries, but also the bronchial circulation was an intimate connection with the plexiform lesion. And later on, and with another study using the state-of-the-art synchrotron-based imaging technology, looking at these plexiform lesions, we identified that the most of the plexiform lesions actually originated from the bronchial circulations. So with these studies, we established that in pulmonary hypertension, bronchial circulation play a critical role. So how does that lead to hypoxia? Well, basically, this is the schematic that explains it. Normally, the pulmonary artery blood should go to the alveolar capillaries. There should be a gas exchange going back to the pulmonary vein and to the heart. However, pulmonary hypertension, we hypothesized that there are these anastomotic connections between the pulmonary artery and the bronchial artery, that the blood basically bypasses the alveolar capillaries and then go into the bronchial microvessels and come back to the pulmonary vein. Therefore, this bypass represents a right to left shunt, and the blood does not get oxygenized, leading to severe hypoxemia. In an animal model, actually these bronchopulmonary anastomosis also exist, which would open our door to study more the regulation of this pathway. So our first hypothesis was that this IBA network is recruited in a Sugen hypoxia pulmonary hypertension rat model. The Sugen hypoxia pH model is well known to the pH field, and the reason why we picked this because this model recapitulate mostly what we see in the lungs of patients with pulmonary hypertension. We established this model in our laboratory. We've proved that these mice develop pulmonary hypertension as well as right ventricle hypertrophy. And then we needed to establish an automated vascular trace injection system that which we did with a syringe injection pump. And with that help, with the steady flow, we were able to entire fill the pulmonary vasculature. In this case, in this picture, you can see that the cannula is in the pulmonary arterial system, and then the vasculature is filled with green ink. Now, so about our preliminary results. Well, first we did the green injection, and we counted and identified how many bronchial vessels are filled or filled at all with green ink. Again, we injecting the the greening into the pulmonary arterial system, which normally is not connected with the bronchial arteries. But in pulmonary hypertension, we hypothesized that these will be open. And sure enough, you can see even after 3 weeks of Sugen hypoxia treatment, there are bronchial vessels that failed with greening. Even more, you can see in 5 weeks of treatment and in 10. This is a typo, I apologize. This is a 10-week Sugen hypoxia treatment. You can see these are much bigger now and more frequent. When we counted these, then you can see that in 3 weeks, the density of the ink-filled bronchial vessels are somewhat increased, but in 5 and 10 weeks, they are significantly increased. And now, not only we can characterize this bronchial circulation involvement by vascular density, but we can also characterize by the size of the shunt. And this is what we did with these experiments. We injected different sized fluorescent microbeads in the pulmonary vasculature of the Sugen hypoxia treated animals. First, we did a 1 to 5 micron ones, as you can see in a low power view, that it goes into the pulmonary arteries and it goes to the capillaries as well, because they're smaller than a normal capillary diameter, which is about 7 to 10 microns. But when we looked at specifically using the standard stains, we identified bronchial vessels. And then when we switched to the immunofluorescent mode, we can identify these beads in the bronchial vessels, which is a pathologic finding. Again, it should not connect with the pulmonary vessel, and a pulmonary flow should not connect with the bronchial circulation. When we use the bigger one, like a 27 micro diameter bead and injected, we also identified very few, but we identified these microbeads in the bronchial vessels. So again, that means that the shunt size, the intrapulmonary bronchopulmonary anastomotic size, is at least 20 to 27 micron based on these observations. And then when we injected the biggest ones, which is a 67 to 750 micron, we only find those in the pulmonary arteries, but not in the bronchial circulations. So in conclusion, we find that the intrapulmonary bronchopulmonary anastomotic network is recruited in the Sugen hypoxia pulmonary hypertension rat model, and then the size of these shunts is at least 20 to 27 micron in diameter. And then we are really excited, because establishing that these networks actually present in an animal model, it opens doors for us to test the regulation, or what type of regulation these anastomotic network are under, and whether we can manipulate these. We're thinking of like closing these shunt pathways to, for example, attenuate the hypoxemia in pulmonary hypertension patients. So thank you very much for attention. I'm ready to answer your questions.
Thank you for bringing us home with that wonderful presentation. I think you addressed one of the questions already, which was what's the potential applicability of this, and sort of what are the treatment implications. I just wanted to follow up on that. Do we have any information about how the current pulmonary hypertension medications affect the bronchial artery anastomoses, and how do you think that could be studied and with our currently available medications?
Right, that is a really relevant question, and really like gives us some headaches over the years, how to sort of dissect, the effect of the pulmonary dilatators or other type of beta blockers or usage of on these. These seem to be regulated by similarly to systemic vessels. So there would be some kind of an overlap when you're using a pulmonary arterial dilatator that would act, the opposite of that. So we need to calculate that. But what we'd like to do is using cell work on bronchial endothelial cells and smooth muscle cells to identify new pathways that we can stay away from the currently used medication, and maybe in with the bronchial or the vascular dilatators like pulmonary dilatators, we can use new medications that targets new pathways on the endothelial cells.
All right, well, wonderful. Well, thanks to everyone for sticking around. I know we ran over time because these talks were so interesting. We couldn't really cut them short. So I just want to again emphasize the commitment of PHA to research. All four of these investigators have, I think, benefited, as you see, from the innovation awards. I just also wanted to mention that we have 3 other awards that the PHA currently funds, which is the Pediatric Research Award, the Early Career Mentor Award, as well as a newer award, which is focusing on healthcare disparities. And so thanks to the PHA for their continued support. And I hope to make this a recurring event through the years, and we'll be excited to see how the research evolves from what you've seen today. So thanks to everyone, and have a great afternoon.
Thank you. Bye.
Thank you.
Thank you.
Thank you, everyone.
Bye-bye.